I will start by just saying briefly, thank you very much for coming this evening. Welcome to our annual symposium public lecture. If you haven't been to one of these before, I'm very glad that you're able to join us tonight. Our goal with these public lectures always follow our annual symposium. We have a keynote speaker come from somewhere else in the country, typically, or at least somewhere outside of UCI, because communicating new ideas and talking about new things that are going on in science and in our labs is such a critical component to advancing science writ large. And tonight we are very pleased to have as our guest special lecturer, Dr. Ali Brivanlou, who joins us from Rockefeller University. Ali obtained his PhD right here in California from University of California, Berkeley, did a postdoc at Harvard, and then started at Rockefeller University for his assistant professor position, moving on up through full professor. He has actually, quite honestly, more awards than I could possibly name. I don't think I have even one, so I'm really quite impressed all the time. And the amazing thing about his work is that it spans this incredible dimension of asking the most basic questions we can imagine in biology. How do we develop? How do you make a human? How do you grow a human? Is that self-organizing? Does it take instruction? How does it happen? And the critical thing to understand there is understanding those processes tell us about stem cells, and it tells us about how we can think about using stem cells and informing stem cells or instructing the stem cells in our bodies to do things better so that we can heal, so that we can do transformative, not just transformative research, but research that is going to help us understand not just human biology, but how do we treat what ails you, so to speak? How do we move out of the bench and into the bedside? And so he's a wonderful example of a scientist who has done not only that, but is able to communicate to all of us and really be able to tell us about his work and convey the excitement of what stem cell and regenerative medicine research means. So with that, I'd like to invite Ali up. He says I'm too kind to him, but it's really not true. He's actually quite wonderful. And ask him to begin his lecture. Thank you very much for joining us.
Wow, again, what a great introduction. This is the second time, and you are wonderful, I have to tell you. It's a pleasure to be here. It's a great honor to be here. I really feel like I'm at home when I'm in Southern California. So I'm very glad that you took the time to come and to listen to what I want to tell you. And hopefully at the end of the evening, we're going to have a different vision about where do we come from? What are the rules that guide the journey that we're in from the beginning till the present and eventually to what we're projecting based on what we know. So I hope you have been enjoying my swimming tadpoles under the moonlight. This is actually the essence of experimental embryology, which is one of the oldest branches in biology. It can be traced back hundreds of years, if not thousands of years. Everybody who ever saw a bird hatching from the egg or a frog egg becoming a tadpole and then a tadpole metamorphosing to become a frog and hopping out of water cannot stop by being mesmerized about this process. There is something naturally attractive about where do things come from and the way things assemble. So these are tadpoles from a frog called Xenopus levis, originally from South Africa, from a very high altitude lake. This frog likes very cold temperature, has a very interesting lifestyle and many different attributes. Among them, the fact that you can generate thousands of embryos on a regular basis from a single female that lays the eggs in pond water. And so that has become an attractive topic for experimental embryologists who needed to have access to biological material in large amounts to be able to just do the basic descriptive work, like when one cell becomes 2, 4, 8, 16, 64, and moves on to create a ball of cells. The continuity of it, the robustness of it has been under scrutiny for many, many years. And I learned that from my great teachers and advisors as I was moving forward in my own education. And the only thing special about these tadpoles is that they're all clones. And so John Gurdon, one of my teachers, got the Nobel Prize for experiments that he did in 1963. And that was the basic understanding of how the genetic information is translated into generating cells and then moving forward toward the generation of organism and evolution of organism. So I'm going to come back to these beautiful tadpoles in a second, and you will see why I'm starting with this. So I am the professor in Laboratory of Synthetic Embryology at the Rockefeller University. The title of my talk in your brochure is a very long title that can be simplified by calling it Human Synthetic Embryos and Organs. And I will define to you what that is. The picture that you're seeing on the screen, along with my title and my position, is a picture of the high-end resolution that we have for imaging an organism as a whole, as opposed to sections or reconstructions. And it tells you really the power of developmental biology in 21st century and how far we have come from the basic understanding of early embryonic development. For full disclosure, I'm not only a professor at the Rockefeller University, but I'm also a co-founder of a startup company called Rumi Scientific. And Rumi is a 13th century mystical Persian poet, and I would be happy to either recite some verses for you at the end or tell you why I'm so attracted by the way he thinks. Okay, so I think a good way to start this story, and I'm going to tell you a story, is by trying to define what is it that when we look at something that evolves in time, this process of self-organization, how does that happen? In non-living systems and the physical world, you're looking at the development of a snowflake. And as you know, a snowflake is made of an assembly of water molecules, and all you have to do is to drop the temperature, and it starts creating these amazingly radially symmetrical structures, each one of them different than the other. And extremely mesmerizing. There doesn't seem to be a program involved in this. It's just some sort of a self-assembly and self-organization that leads to these magical and complex structures. And each snowflake, of course, is different than the other one. And every time we get a drop of rain in our head, it starts as a structure like this. So in non-living physical world, self-organization and self-assembly is the norm, and we see it all around us. This is the infinitely small scale or relatively infinitely small scale. You see the same thing in larger scale of non-living systems. This is the formation of the galaxy. You notice that there is always a center in these events, and things are organizing around the center, most of the time in an inside to outside manner, and there is a very strong dynamic that's associated with this kind of organization. And we're not going to go to the details, but there is a difference between self-assembly and self-organization. In biological systems, you have examples of both. So and this is a non-living system in different scales. In biological systems, in microscopic scales, you're looking at the assembly of a phage or a virus, a bacterial virus, where you can take the components apart, the different proteins that make the capsid or the inner components. You can put them in different tubes, and then you can recombine the tubes, and this thing self-organizes and self-assembles to generate something that's much more than the sum of the parts. In living system, this happens not only in microscopic scales, but of course you have seen it many times in nature, in all aspects that you're looking at. These are a flock of birds somewhere in Arizona, and you can see that the structures, the dynamic structures that are being generated are much bigger than the individual components of it. To a large extent, I can consider this as a superorganism, if you consider each bird to be a cell of something that's much bigger than the individual. Okay, but among all the, I think, most attractive self-organization system that I know, and again, I'm biased, is this one. This is a frog embryo again, and I have lined up 10 of them for you in this slide. And these embryos develop in pond water. So they're sitting there. This movie was made actually by my two boys when they were doing an internship in my lab. They asked me what experiment to do, and I said, just look at this and film them. And you will see why this is interesting. And I could save one of them from not going into science, but the other one is already deep into science. So this is why I think this is mesmerizing. There is no information coming from outside in the system, but if I launch this movie, you will see something pretty amazing going on very fast. At the beginning, it's kind of fuzzy. It's not exactly clear what's going on, But very quickly, you start seeing that the sphere is changing shape. So then you start recognizing the contour of a tadpole, the head, the tail, the trunk, the individuality, the formation of individual tadpoles within that assembly. And again, all of this complexity comes from information that's inside of that one egg. There is no information coming from outside, and yet you generate something that is made of 40 million cells from a single cell by just reutilizing the information that exists in a manner that's very similar to using the keyboard of a piano to generate as many harmonies or music and symphony that you want to with a limited set of components, including the fact of the silence between the nodes. So within a given set of genetic information in DNA sequence, you can generate infinite number of forms and shapes. And I think that's one of the most amazing example to me about how complexity can arise from one cell to 40 million cells, in the case of the frog, in 48 hours. So this is faster than a bacteria growing in a culture dish, if you want to use an analogy about how fast things happen. That means that in every moment, as time moves forward, the cells have to make precise decisions, not only about their own destiny and their own fate, but the way this fate and establishment of patterns are coordinated with their neighbors, and the way you generate a tadpole that's consistently the same over and over again, and that occurs over hundreds of millions of years. Okay, so how does this magical process happen? How do we start generating this complexity? Well, there are lessons that we learn from experimental embryology in a very traditional sense. This is at a time when the frog egg is made of about 64 cells, maybe 128 cells, and you can recognize them there. And there was a big question in 18th, 19th century by just observing these structures as to how does a given cell in this ball of cells, how does she know who she's going to be? What are the decisions about, am I going to be a brain cell, muscle, blood, or bone come from? Seemingly, when you look at them, you can see differences in pigments and coloration, but otherwise, it would be very difficult to predict who's going to become what. And many different schools of thought came to this approach from many different angles. Embryology, again, being a very old school, is also affected by tradition, history, and social expressions. So the big schools of embryology include the German school of embryology, the Japanese school of embryology, the British school, more recently the American school, all aiming to answer the same question. Like, well, how do you generate fate? How do you establish this different kind of cell type within a ball of cell? And so there were big theories. At the end of 19th century, the British theory was dominant. And the idea was cells know who they are based on their lineage. So if you know who your grandpa and your grandma is, that defines you. It's very British to a large extent. That is, if you're an aristocrat, then you're likely to be an aristocrat. And if you're at the flag and you're a blue-collar worker, it's likely that you're going to end up being a blue-collar worker. And so this was a lineage fake determination, like you know who you are based on your genealogy and your descendant. The German school started challenging that in the early 1920s, 1924, by suggesting that perhaps it's not necessarily lineage, because things can move and change. For example, if you pluck one cell from one of the positions here, let's say this one, and you transfer it to a different position, within a window of time, that cell starts following something else that she would have done otherwise. Okay, so this debate continued for many, many years, decades and decades of discussions. And then here comes the American school and clearly demonstrates the series of experiments that I will be happy to describe to you, that this is actually the neighborhood that establishes your fate, not your lineage. And that's very American too. So if you're within a given neighborhood, you're likely to become X, Y, and Z, and if you're in a different neighborhood, you become something else. So this debate was going on for a long time, and I'm going to make a very long story short. You know who you are based on the environment in which you find yourselves. So this is called inductive interactions. At any given time, a given cell receives information from its neighbors and then exchanges information with the neighbors in a very, very fast way. It's as if I were talking to you in 1,000 words a second. And so the cell cycle is about 20 minutes. That means a cell has 20 minutes to decide what direction she's going to go. And if you do a back of the envelope calculation, the minimum amount of time that you need to replicate your DNA is about 20 minutes. So as the cells are dividing, they have to make these decisions very quickly. They have to make it very accurately. And if they're wrong or out of sync, you generate a tadpole with 2 heads or 5 tails and forms that you don't want to necessarily end up with if you want evolution to select you in moving forward. So that's one of the lessons of classical, traditional experimental embryology that is sustained now, and I will show you to what extent this is applicable to our own development in a second. The second principle that emerged from this was this thing called symmetry breaking. You notice that this is a ball of cell. It has a cylindrical symmetry. It's a little bit like planet Earth. I can make an axis between north and south. I can rotate, and things will be constant around that one axis. You can have differences at the surface, but the axis of symmetry is only one. In development, you start with the cylindrical symmetry of a single axis, or spherical symmetry, and then suddenly things become different in one side versus the other side. This is the same embryo in about 4 hours later. There's many more cells than this because things are happening very fast, but then suddenly one side of the embryo is very different than the other side. So this symmetry breaking event is a key determinant factor. You cannot screw this up as you move forward as an embryo because there are consequences. And so this happens in different stages. The fate determination occurs during this stage called blastula, and the symmetry breaking occurs in this stage called gastrula. And these are historical terms that have been used over and over again in many different species. Okay, so if you look at the consequence of that symmetry breaking, this is what you see. You make a sphere to become elongated. Now you have bilateral symmetry with 3 different axes: the head, the tail, the dorsal side, the ventral side, and right and left. So you break one axis into 3 axes, and magically, this ball of cells suddenly becomes something that you can recognize as an individual that's going to move forward. And we know a lot about this. And I spent 25 years of my life trying to crack the code. And how do you get from here to here? And I can assure you that it was a very pleasant journey. It was very interesting. It was relatively easy compared to what was ahead. And what was ahead was, to what extent do you believe that this kind of self-organization occurs in our own development, in human development? Is this something that's evolutionarily conserved, or somehow our embryogenesis is different in ways that model systems such as the frog or the mouse or the bird cannot recapitulate? After all, we're human beings. We consider ourselves much more sophisticated than a tadpole. So maybe things are a little bit different, and maybe we have activities that cannot be somehow imaged or understood by looking at other systems. So I gave this talk this morning. Some of the slides I'm going to show you today might be redundant to those who are in my audience this morning, but it's very important for me to make this point that this is you. You're looking at yourself. This is the fertilized egg. This is this structure called a blastula. This is the same as what we had in the frog, the ball of cells, except that for us, we're a ball of cells, but made more of a soccer ball with a tennis ball inside. The tennis ball gives rise to the entire embryo. The soccer ball outside gives rise to all the extraembryonic tissue. Okay, so single axis of symmetry, everything is spherical in this, and then this magic moment of symmetry breaking during this stage called gastrula, just like in the frog, where when you look on top of that tennis ball, this is what you see. Suddenly one side of it is different than the other side. It looks a little bit like a volcano with a crater, and the crater is broken in one side versus the other side. The process of symmetry breaking mimics exactly what is going on in all other animals, except that, of course, we don't grow in pond water. We grow in an environment of the maternal fluid in which we find ourselves during early development. About 3 weeks later, this is what you look like from a dorsal view. The brain is still open. The spinal cord is zipping itself down, closing the neural tube. And about 1 month later, this is how beautiful you are, including with your gorgeous tail and your gills. And if I had to stretch this for you from one end to the other and make it a line, it would be really similar to a tadpole. And not only in terms of course, speed is different, timing is different, it takes much longer, but in terms of architecture and morphogenesis, it's not that different. So it almost becomes intuitively obvious that principles are evolutionarily conserved. There was a lot of theories about what the presence of this tail or the gills actually mean. There were theories about maybe development is recapitulating evolution. Maybe we start in the womb first as a fish, then the process of birth is like the metamorphosis, where the tadpole turns into a frog and comes out from the liquid environment to breathe in the air, catching the first breath, and then things continue. It's difficult to draw this analogy, and I'd be happy to speculate about it if you want, but certainly it doesn't escape your attention that a lot of interesting correlation between model systems and humans. Two things to remember. One is that the early decision about the body axis, anterior, posterior, dorsal, ventral, right, left, happened very, very early, at the time where women don't even know they're pregnant. So all this very important decision about cell lineage and who's going to be what occurs in the womb without any external conscious knowledge of the events happening. The second thing is a quote that I love from one of the greatest embryologists in contemporary world, Lewis Wolpert, that we lost last year. He said something that I think I always remind my students, and sometimes myself. He says, it's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And Ray Keller, one of my teachers, also John Gurdon, I was puzzled by this. Like, why is it, go through a lot of things. I'm an immigrant and a refugee from the Middle East, and I've seen a lot of big moments in my life, and I can assure you that gastrulation was not part of my list. And it told me, well, you don't understand, because if you have a problem during gastrulation, you cannot separate your head from your butt. So yeah, you reminded me that some of our politicians might have problems during gastrulation. But anyway, this is really serious. This is the time where a lot of important decisions are being made. And we don't want to screw this up. We don't want to miss it. We have to keep with the program, and things are under self-organizing principle moving very fast. So it was a big black box. If women don't know they're pregnant, how do I study this? How do I even collect the samples to study it? What are the ethical and moral boundaries, the social boundaries behind these kind of studies? This is not going to be a frog system or a fish system where I can have abundance of thousands of eggs in my hand and go and figure out exactly who's talking to whom, at what frequency and what speed and what kind of language. To design some sort of way to at least see it with my eyes first, and then try to model it if I can. So a few years ago, my lab developed a technology where you can take that ball of cell and attach it to a substrate and fully get as if it was attached to the uterus. So for the first time, we saw with our own eyes the process of a human embryo attaching to a substrate and the beginning of symmetry breaking, how a ball of cell starts undergoing gastrulation. And what I'm going to show you is what is inside of this structure. This is 12 days old. There are about 1,000 cells. And what you see here in green is the tennis ball. The rest of it is the soccer ball. The way attachment happens is that the tennis ball attaches to maternal tissue, and then the soccer ball lands on it like a parachute. So you generate almost this disc starting from a sphere, and that's the beginning of breaking symmetry. I'm going to put you in a little drone and give you a tour and take you inside of that structure so you appreciate the beauty of it. This is the genetic code and the markers. It doesn't matter what they are. This is the tennis ball. You see two cavities in here. These are the signature of the human embryo cavities. You can see that there is some thickness, but also what you see immediately and appreciate immediately is that there is some sort of a radial symmetry with the center and a group of cells around. These markers, these molecular signatures that are color-coded here, are the signature of cell types. So the way we recognize cells is not by morphology. Just like in the frog, I told you, it's very difficult to know who's going to be what just by looking at them, but it comes with a genetic signature, like a fingerprint. That's how you know what kind of a cell you're looking at, and that's how you can follow and track it. So that was cool. That was the first time we could see ourselves at this stage of development. But at the same time, like everything else in science, the minute you start solving a question, you raise another 1,000 questions. So why is this useful? How do you justify doing this? How far are you going to go if you're going to do this? And that's a very important question that transcends basic scrutiny and scientific studies to include everything that we consider as what is acceptable or not, depending on the culture and the history from which we come from. But one of the clear benefits of this is that you start seeing things by definition that nobody else has seen before. Species-specific trait. This one example of a human cell type that was never seen in any other mammalian species before. And that provides a rationale as to why is it that if we really want to be serious about understanding our own development, by definition, we'll have to look in our own species as opposed to trying to extrapolate from others. Okay, so today I'm going to spend 5 minutes in 4 different topics with you to give you a little glimpse about where we are, where we're trying to go, and why do we find ourselves where we are, or we think where we are. The first one is to accept the fact that we're never going to have enough embryos. Therefore, we need to generate models. We need to generate models that mimic the embryo. Now that we can see even 1 or 2 real embryos, can we actually use a system to generate thousands of them without having to go to the impossibility of collecting natural samples? So I'm going to tell you about this thing called synthetic embryos. They're embryo models. I'm going to tell you about gastruloids that model gastrulation. I'm going to tell you about cerebroids, neuroids that model the formation of the brain and the nervous system. I'm going to give you another example that we call lungoids. This is for lungs, and you will see why this is interesting to generate tens of thousands of human lungs. You can already guess. I'm going to tell you a utilitarian aspect of doing this kind of work, which is disease modeling. I'm going to give you 2 examples, neurodegeneration by focusing 2 or 3 slides on Huntington disease. I'm going to give you an example of infection by showing you SARS-CoV-2 progression in human lungs. And then I'm going to detach a little bit from science to tell you the consequences about doing this kind of work, these structures that I call infernoid. And this is me, upside down, going into this hot oil. Dante would have pictured me like that. So let's start from this now that the rationale is established. How do we do this? Well, we start with human embryonic stem cells. As you know, human embryonic stem cells are derived from the early human embryo. This is the tennis ball. This is the soccer ball. You can pluck one of these cells out, put it in culture, and this cell represented as a white circle here is called an embryonic stem cell. It has this very interesting property called totipotency. That means that it can generate all the cell types of the human body. So there are 2 magical properties about these cells. One is that it can self-renew. You can keep these things in culture forever, decades and decades, and you can keep them frozen in time. So they will not change fate. They're always going to say the same character as they were when they come out of the tennis ball. So that's called self-renewal. It maintains itself forever. The second magical property that no other stem cell has, there are many different kinds of stem cells, but embryonic stem cells have this other magical property where they can give rise to every single cell type of the body. If the cells, the different organs or cells are represented by these different colors, this is the only cell that can generate all of them, without exception. The only other cell that we know in the universe that can do that is a fertilized egg. So here you have a system where you can have infinite access to cells that have all the information necessary and sufficient to generate the whole organism. In fact, in model systems like the mouse, if you do the same experiment, you can generate an entire mouse from a single cell. So I think this is magical and is absolutely fascinating. In model systems, of course, the way you prove that is that you take one of these cells and you put it inside of an embryo and you mark it, and you can see that the progeny of these cells have contributed to all the aspects of the organs and the adults. But how would we do that in humans? It's really a functional definition. It's obviously impossible to imagine that you're going to take this and put it back to a human blastocyst and implant that and then look at the other end to see where the progenies are contributing to. It's out of the question. So how do we solve this? This is already, or in year 2000, 2001, in the lab, and this functional definition had to be tested somehow. We call these cells, because they were derived at Rockefeller, the Rockefeller University Human Embryonic Stem Cell Collection, for short is RUES. And these lines, despite the controversy, and I'm going to show you a couple of examples at the end during the infernoid part, that we had to define a way to functionally test them to make sure that they can do that. And of course, we cannot do that in humans. So we suggested some time ago, putting a lot of the people in the world that I respect, my own teachers, advisors, and people who are really willing to move forward into uncharted territories, that perhaps one of the best way to test that functionally is to transplant a human cell into a mouse embryo and then ask, can this human cell contribute to different mouse organs? And so this proposal was published as a white paper in 2003. And to make a very long story short, after about 2.5 years of trying, we generated for the first time in 2006 a mouse embryo. That's a 14-day-old mouse embryo. Mouse gestation, by the way, is 21 days, much faster than humans. And you can see here that the human cells that are represented in red have contributed to all the organs or all the derivatives of all the germ layers of the organs of a mouse, eyes, brain structure, spinal cord, heart, limbs, and everything else. So that was the first time, and people call this chimeras or hybrid based on the historical definition of mixed animals. But this was the first evidence that human embryonic stem cells can functionally contribute to all the organs. And of course, you can imagine that was extremely controversial, but that's what it took. 189 attempts before we got one, and we realized that this would be impossible. But that one, just like the in vitro attached embryo, was sufficient for us to conclude that this is functionally valid. Okay, now that we have this, what do we do with it? Well, we put the cells in culture. We create colonies. These are colonies. Each single white dot is a cell. They create colonies of different shapes. And we realized it's very difficult to quantify and to follow and track. So we decided to impose architecture in this. We generated this culture called macro patterns. So this is a very small glass chip where you force the cells to grow in a confined geometry, in this case, circular geometry. So we impose the architecture and we seed the cells and they grow in different sizes in these circles, the largest one being 1,000 micron, the smallest one being 85 micron. And then we develop tools, deep neural net and artificial intelligence networks to track every single cell in every single colony in real time. So now this gives us the power to, just like the frog, where the video that I showed you about how do you make a tadpole out of a fertilized egg, that gives us the first glimpse as to how do you make a human gastrula out of a cell that mimics or actually models an egg. And so by doing that, we follow what we thought we knew from frogs and mouse and birds. And sure enough, despite, to our big surprise, and despite any expectations, we saw that with a single signal, in this case, the signal is called BMP4. It doesn't really matter what it is. But in response to a single signal, suddenly that each circle starts self-organizing with the center cells demarcated here in blue by a gene that's called ectoderm. It's the great-grandfather of your nervous system and skin and derivatives such as that, including your brain. Surrounding it, this group of cells in red, that's the great-great-grandmother of your blood, muscle, bone, and cartilage. And outside of that, every single cell type that we know and we recognize with molecular markers, including endoderm, including germ cells, RUES2, this cell line is an XX female cell, so the germ cells in red here are the great-great-grandmothers of the oocytes and the eggs. And we had control, that gives us control about geometry, it gave us control about dynamics, it gave us control in, of course, modifying genetic using genomic editing tools, CRISPR-Cas9 and others, and now we have the tools to finally dive into the principle of self-organization that involves our own development. And that was published over a long series of papers. I'm not going to go to details. If you're interested about the mechanics behind it, I will gladly articulate that for you. The reason these two markers were jumping here is because we're very excited to see them. This is the signature of gastrulation. So this is the first time we see human gastrulation with our own eyes. It's like a mirror reflecting our past. We can see what happened before we became as sophisticated as everybody sitting in that audience. You can also track this dynamically. In this case, you can tag every cell with a different color This is the code for the colors. But one thing you're going to notice when I launch this movie is that self-organization in human gastrula or human gastrula models occurs from outside in. So everybody is blue at the beginning. At the edge, blue is gone, red shows up, and at the edge of red, red is turned off and yellow shows up. And this moves from a manner from outside to inside. So this self-organization is the reverse of a snowflake, or the larger dimensions I showed you from inside to out. There is something very magical about this, is that it is self-organizing, but it is self-organizing from outside to inside in an inward fashion. So that means that every single cell, and that's a technical thing that I'm not going to take you, can measure its distance from the edge, and the way she knows who she is to figure out how far away from the edge she is. The farthest distance would make her blue, the nearer distance would make her yellow. And so the nature of this ruler was clarified over a series of 6 years, 7 years of studies. It's actually relatively simple ruler that the cells use. There's a physical edge sensing mechanism and then a reaction diffusion mechanism. And these are technical terms to tell you that the cells sense in a different way the signals one are at the edge versus at the center, and I'll be happy to go to the details if you're interested. But I don't think that this is the point for the audience I'm addressing today. How do we functionally test this? How do we know that we're not looking at something that can happen in a glass microchip and might be relevant to our own reality of development? Well, one way to do this is to make an attempt to grow these structures in an embryonic ecosystem. And again, of course, we're not going to do this in the context of humans or even primates. So we develop a technology that's based on very old traditional classical embryology, dating back to Nicole Le Douarin in the 1950s in National Academy of Sciences in France, all the way to experiments done by Hamilton in the US in the 1930s, which is basically an engrafting experiment. You take a piece from one embryo and you engraft it to another embryo and you track it and to ask how does it behave. So we did that with the chick system. We decided that, and as I told you, the mouse was very difficult, 189 attempts to get one that was successful. One of my very talented postdoc, Tati Cano, joined the lab from Brazil. She was an expert in bird embryogenesis. And she decided to take these human synthetic models, peel them up from the chip, transplant it inside of a bird egg, right around the yolk, and then opening up a window and then closing the window and putting in the incubator. Eggs, bird eggs and chicken eggs are cheap. You can grow them easily. They develop very fast, and better than 89%, 90% of the experiment were extremely successful in generating this ectopic structure inside of the egg. This is the bird itself with the head and the backbone. Here you can recognize it on the shadow. This is the human embryo that's been transplanted, the model embryo, side by side. And you can see it makes its own territory, and symmetry is broken. You go from a circle to a line, and you can trace it because you have your CRISPR-Cas9 genetic markers in that. And that's mind-blowing to think about, that this is even possible. If you consider that birds are closer to dinosaurs than they are to humans, that experiment already tells you that whatever principle of the ecosystem is that maintains and allows differentiation of embryonic cells have been conserved over more than 500 million years. And again, that is in line with thinking that embryogenesis follows a set of rules that were established long time ago in evolutionary adaptation in all species, or at least invertebrate species. I'm going to put you in a drone again. I'm going to take you inside of this structure that I represent here. The human cells are in color. This is a marker for all the nuclei, DAPI in gray. And if I take you around this, you will see the landscape is a bit hilly and bumpy, but you can see clearly that the human models have actually broken symmetry. They generate an anterior and posterior axis. These are optical sections. When I slice this with light, so you can see what's inside, human cells are nicely docked underneath the bird. If I pull you out, you will see and you will recognize some of the structures I showed you from the early human embryo pictures. This is the dorsal side with the spinal cord, the brain that's open on the anterior part, and the cells that are moving on each side. So you have anterior, posterior, right, left, and then the dorsal ventral when you cross-section that. And so there is a lot of amazing things in this experiment that can be interpreted in so many different ways. First of all, the speed of development. You could have already seen that in the context of the mouse. Human cells can be accelerated to differentiate in different subtypes at speeds that are beyond belief, 100 times more than the natural environment. That means the ability to differentiate to an organ is not limited by the cell cycle or by other components. It's directly influenced by the ecosystem in which you find yourself. This is an interesting topic of timing of differentiation that my lab is interested in. But beyond that, it really gave us an opportunity to detach from the glass ship, literally, and ask about the potential of the humans in development within an embryonic environment. Okay, so that was the first example of the human bird camera. It made the situation much easier than the human mouse, and the symmetry breaking for the first time to humans. So now we're moving almost from 2D on a glass sheet to something that's closer to 3D. Can we learn from this and recapitulate this third dimension in the human system in vitro in a culture dish? And the answer is yes. This is the work of Miho Simonovic. And he showed that by creating an environment with gels and stiffness and elasticity and viscosity, he came from the theoretical physics world and measured very carefully the environment in which a human embryo is naturally, to the best that we can estimate, naturally growing. And we can recognize in these structures that you can generate by thousands now the two cavities of the embryo, and again, the exact molecular signature of what you would see in the natural embryo. This is the one I showed you earlier when I put you in the drone going inside of the in vitro attached embryo. You recognize the two cavities, the same molecular markers, and the same signatures. And then if you allow that to grow further, it will actually break symmetry where one side of the embryo will be different than the other side. So all those things are self-organizing in a context outside of the womb, outside of the uterus, and they're moving forward. It is actually amazing to see the speed at which this progression is happening. This is a natural embryo, meaning the product of a sperm and an egg, a fertilized egg, grown for 8 days, and the synthetic 3D embryos coming from embryonic stem cells. And you can see that structurally, at least by regular optics, they look very, very similar. In fact, if you stick a pregnancy test in your Petri dish, they will score positive. So this is, I think, one of the most functional aspects that self-organization of human embryos can be modeled in a way, and again, by numbers that are very similar to model systems. And therefore, we finally have a platform where we can really carefully understand the program that's underlying the formation of the brain or blood or muscle or bone or cartilage. Okay, but now we have a problem here. There was this rule set up early on in the 1970s when IVF was created. So there was this big deal. Maybe some of you remember, we're making test tube babies, we're all gonna die. There will be Nazi troops walking on First Avenue in New York. And what are we doing? We need to have some sort of a regulation. It's all fair and perfect. And somehow there was a limit drawn at day 14. You can only work on the human embryo, natural human embryo, up to 14 days. After that, there was a line on the sand. You could not cross that line. And it was based on a series of considerations, scientific and others, that kind of were linked to the interpretation of individuality and symmetry breaking and other components, which I would be happy to describe more in detail if you're interested. But now we're dealing with a system where the cells have been in culture for almost 20 years now, and we reprogram them to move forward and generate an embryo. So how do I count time? When we say 14 days, T0, the clock starts at fertilization and then moves forward. If I'm starting with a cell line that has been in culture for decades, I don't have a T0, so I don't know where I am in time. And that became something that had to be solved, and at the end, I will show you a possible solution to it. Meanwhile, I'm going to take you in the next few slides quickly to organs, and my favorite one being the brain and the cranial structures. If I can make an embryo, we should be able to make brain with things called cerebroid, neuroids, which are craniofacial structures, and as I said, perhaps other tissues. So this is the collection of tissues we have made so far, the embryo I described to you, and I'm going to describe these two very quickly in 2 slides, and then I'm going to take you to the last ones. So this is the macrochip that I showed you, but this time rather than self-organizing human embryos, we're self-organizing human brains. They have radial symmetry. This is the molecular markers defining the different cell type. You recognize the center and the edge again. And if you ask, where are we in our timing of development? The answer is we're somewhere in the 3rd week of development. And if you ask, where are we? We're in the most anterior part of the open brain. This is uncharted territory. So we're walking into a space that no one else has walked before. And we, of course, expect and we do see things that nobody else has seen before. If you look at these structures, space filling programs, anatomically, they are really macro brains. We call them brains on chips because they have not only the morphology and the molecular signature, but also as a tissue they assemble in ways that are very similar to a fetal brain. This is a craniofacial structure that we call neuriloids. This doesn't matter. This is the protocol as to how to get here, and these are, again, the molecular markers. But again, you generate everything that is the brain in the center in green, and then these things called neural crests that make the craniofacial structures, and then the sensory organs, eyes, olfactory, and others, and all covered with skin as demarcated by these markers. And again, we find ourselves in the same time of development, but this time around in a little bit more posterior structure than we were with the brain. Okay. So we can do all of these things. We can play these games forever. You name the tissue you want, and we should be able to self-organize it. And you can imagine the applications for that for regenerative medicine and others. So that comes the question. And usually the reaction is to this is this knowledge for the sake of knowledge, or is there something else in there that can be useful? Certainly when you talk about human embryos and human stem cells, you need more than just basic science information. So one way to demonstrate the power of this system is to use them as a model to actually do disease, representation of disease in humans that otherwise could have not been addressed by any other systems. So I'm going to give you 2 quick examples, one with modeling Huntington disease in human embryos. And so this is the property of Huntington. It's a single mutation in a single gene. It's considered a neurodegenerative disease. That means after the nervous system is being made. And there is no therapy or cure or any approach that can help that, unfortunately. So if you do have the mutation, you're out of luck. And so that's 100% penetrance. And the reason for this is not that somehow neuroscientists are not as smart as others, but the reason is that you cannot model these systems in mouse and in animals. So this was supported by the fact of discovering a gene for Huntington that is only expressed in hominids, that's high-end non-human primates and humans, that established the fact that this, to be serious to study, needs to be studied in human, cannot be modeled in the mouse. And so we used the gastrulloid system to generate the mutation by using CRISPR-Cas9. So now we have 2 identical lines, one with Huntington and one without Huntington. And sure enough, when you do this, this is the signature of self-organization in a non-Huntington case. And if you look at a single mutation in cells that are otherwise identical in their genome, you get a different signature. So this signature you can see has a smaller center and a wider edge. And if you do this with the cerebroids, you will see something very similar. You can quantify that again with tools with subcellular resolution. Neurilo is the same, specific signature for the disease. So for the first time, that allowed a couple of things to happen. First of all, the suggestion that Huntington disease is not a neurodegenerative disease, but is actually a developmental disease. I'll be happy to expand on that. Second, that Huntington disease does not represent somehow a gain of toxic function, but we demonstrated that it does represent a loss of function. When you have a mutation, you actually lose an activity. You don't gain or modify something that already exists. And then finally, being able to model a human disease in a human system rather than in a model system. So one of the mission of Rumi is to take this technology in a high throughput screen. So we use robotics to put these neuraloids in small wells. We use our deep neural net and artificial intelligence for phenotypic analysis. And the good news is, and this is for Francis, I think the good news is that if you use that system on isogenic lines, you can actually, so this is the signature of a Rockefeller University or US2 lines, on a neurilloid. This is the signature when you have the Huntington mutation, and you can see night and day different. And if you use this as a platform to screen for drugs that would revert this signature back to a non-HD, you land every once in a while in a compound that can rescue or save this back to this. This is much more similar than it is to this. And that was the good news for the first time. And these compounds can be identified using a model system. Interestingly, there are many foundations that support Huntington disease research, CHDI included. And once they see these kind of results, they realize that perhaps modeling on humans is the only way if we're going to be serious moving forward on that. Okay, so this is brain. Here's another one that you might like even a little bit more. This is the formation of lung buds or longoids from the macro patterns. This is what we generate. The structures look already with broken symmetry. The tip end of it is different than the attached end of it. They really mimic the human fetal lungs that are represented. This is natural human tissue. And they can use molecular markers to see that they are self-organizing to make synthetic lungs or lung models. And for the first time, we can mass produce this in the microchips by tens of thousands that are genetically identical. So we started this experiment second month of COVID when things were getting shut down. And Charlie Rice, we started this experiment before Charlie got the Nobel Prize. He ended up getting the Nobel Prize 2 years later. But he came, and the great thing about Rockefeller is this very close inductive interactions among small faculty number. And so he came to us and said, the number one limitation to actually try to deal with the virus in a serious way is to obtain tissue that can be infected and tracked in real time. The only base almost 3 years ago, 2.5 years ago, was primary tissue from patients. And of course, there is variation from one patient to the other. It's impossible to maintain and to reproduce, or even less to quantify. So this was the rationale behind the project when we started this. And at the time, I think you guys all remember, it was called the invisible enemy because it was impossible to track. So in collaboration with Charlie's team, my team used these lungs to infect with virus. The virus is in blue. There is these molecular markers for the lung. And as you can see, the virus can infect these lungs directly on a microchip. And the infection is very specific to one end of the lung and much less to the other. It's always to the proximal rather than distal side. And there are very specific cell types sitting there as opposed to the other end. And that was the beginning of tracking the virus in real time. And this is one example of it. Again, I'm going to take you to this Z-Strack. This is one of those lung buds, and each blue dot is the virus. So now the invisible enemy is visible, and they can start playing games with it. For example, which vaccine is better, or what monoclonal antibody is better in neutralizing. And so by doing that systematically, this paper is in press, and it's going to come out hopefully soon. Showed to compare different monoclonal antibodies. This one is from Regeneron, the C121 antibodies. Of course, everybody thinks that their antibodies and vaccine is better than others, but there was no quantitative platform to test that directly. And here's one example of one of them, and there are others. And C121 is the one that was actually used for President Trump for the infection. Okay, so the application of this technology is moving forward in many different directions. People interested in kidney disease, in pancreatic, even cancer modeling and self-organizing structures are converging, and we gladly provide. We cannot do, obviously, as an academic lab. Rumi is still very small as a startup company. So collaboration with people like Charlie and others are routine for us, and we welcome that very much. Okay, so I'm going to end with the horizon. Where are we going really with this, and how far do we want to go? What is the boundary between what is possible and what we should do? So this is back to my flock of birds. A couple of things that are in progress. First of all, I call this comparative human anatomy. So we generate chips with different shapes and form. I showed you the circle. You can make all kinds of things. You can make triangles, you can make Pac-Mans, you can make circles within circles. The reason you do this is you appreciate the way the cells actually measure the distance. It's mind-blowing to think that a human embryonic stem cell can actually tell if it's a line or if it's a curve that is facing it from a distance. Not only it can tell if it's a line, it can actually read the angle. So you can say if it's a sharp or a wide angle. These kind of measurements of appreciation of contours and architectural boundaries was extremely unexpected to me and to my physics colleagues who love to do mathematical modeling. Now, you have to be careful with mathematical modeling because those guys like to go a little bit too far on the theory and detach from reality. It becomes more like what does a cell can do versus what it does. And a lot of biology sometimes is diverted toward that direction. So we're moving forward with this to understand the aspect of cells recognizing their environment and cells recognizing the boundary about what is a neighbor versus what is an edge. We're also moving forward to detach from chemical stimulation. Everything that I showed you was based on adding a chemical substance to a colony and then creating self-organization, like turning on a chemical pathway. But there are physical ways of self-organizing structures. You know, in many different fungi and in plants, you can interpret physical stimulus in order to generate biochemical outcome or biological structures and forms. A sunflower would always orient toward where the light is. Photosynthesis uses, uses natural light to generate biochemical pathway and energy. So we ask, can we generate a system in human embryonic stem cells where the human cells would be responsive to light, no longer to chemicals in order to self-organize? So again, with the physics colleagues, the same that collaborated with us to generate the macro pattern in the cover slip, we generate a system where you can actually generate specific lights at different wavelengths in different shades on a plate of cells. So this allows us for the first time to send a signal where the human cell acts as if it was undergoing photosynthesis and self-organizes in dynamic range. I think this is, to me, one of the most fabulous positive outcome of this kind of work, is that generating shape and fate based on physical stimulation. Imagine a day where needles will not be relevant if you need an insulin shot, and you can just shine light to a patch that attach you. And I think this is the way we move forward: self-organization of human embryos and structures using physical stimulation rather than chemical stimulation. You can apply this technology directly to the macro pattern. So if you use a single circle macro pattern, you can actually shine light in different kind of windows at the given positions. In this case, at 9 o'clock. It can be at 12, 3, or 6. You can shine. You can break symmetry by doing that in a way that the embryo does. So suddenly, this side of the embryo is different than the other side. So you're breaking symmetry by using a physical light stimulation as opposed to using a chemical activation. Okay, so there are boundaries about how far we can go with this and why we should. And this created a lot of issues. There is a society called ISSCR, some of them might be familiar with, is International Society for Stem Cell Research. And when this technology was moving forward, we're clearly past day 14 in our modeling. So there was a big discussion that was driven by scientists, ethicists, social scientists, members of different backgrounds and cultures. And there was 14 different countries engaged in more than 100 Zoom calls. That was the entire time of COVID. And you can imagine what it takes to put all these people together and create these Zoom calls in different time zones, but basically to address what is okay and what is not okay, and how far. It's a little bit what happened in the 1970s with the IVF. So basically, I can summarize the conclusions in the 4 points that you're seeing on the screen. First of all, the 14-day rule vanished. It became very clear that the definition of that boundary needs to be revised because the model's already passed that stage, and we need to make a decision as to how far. I pushed for that personally for reasons that to me are extremely justified, and I think the best way to deal with it is the utilitarian aspect of the argument, that if you're going to be serious about dealing with developmental diseases, you need to use developmental models to do that. Otherwise, we're not going to move forward. The second thing is that generating embryo models are okay. You might not believe, but there was a tremendous objection about creating model embryos in a microchip. For example, do those gastroid, should they be endowed of rights or moral values? And so just to explain that these are cells in culture, and cells have been in culture since the 1950s, including human fetal cells. They have been the basis of generation of vaccines and treatments. in so many different ways, in so many different platforms, that just by putting a contour or an edge to this cell culture does not qualify them as superior moral or ethical entity than regular cell culture. That was a little bit unexpectedly difficult for me. That making chimeras was okay, as long as you don't use a human to non-human primate, which we all accept 100%, but generating a model human embryo inside of a bird egg, this is not something that the society needs to necessarily get shocked or insulted by it. And one thing that we could use arguments for this among all of us from all these different countries to agree on that. But one thing that became really a subject of contention, and I couldn't make that argument, unfortunately, at least for now until the next round, is that we are able to make human germ cells from embryonic stem cells. As I said, embryonic stem cells can make all cell types. This is an RUS2 background. So I show you on the next slide that we can make germ cells. These are called primordial germ cells. And it's tremendous use in IVF. As you all know, sperm is cheap, but eggs are very precious. So one of the most limitations of IVF is women's egg in terms of quality and quantity decline with time. And as society is accommodating more and more equality among genders, this becomes really an issue, a social issue that needs to be addressed. And just like for the human embryonic stem cells, you need a functional assay to do that. So we can generate the great-grandmother of eggs and oocytes in a microchip by the thousands. You can even generate it from iPSC. So in a patient-specific, in a person-specific manner, you can generate an infinite amount of germ cells. And you can actually look at their structures and compare them with the situation in vivo, just like I showed you for the lungs and comparing the lung ovaries. These are the natural ovaries developing with the different markers. You can recognize radial symmetry with the center and this beautiful thing sitting in the middle of it, just like a nest that's going to become the next oocyte and mature to become an egg. And you can generate this in the macro patterns, and you can create conditions where on a given size of macro pattern, you recreate these ovaries from scratch. So this is one of these colonies here, and you're looking at the structures. I'm putting you again inside of the drone to show you what's in it, and the structure and the morphology is very similar. Markers are different colors, but you appreciate the architecture. So if this was to be a real thing, it has to be detached again from morphology and from molecular signature. It needs to be functionally tested. The definition of an egg is a cell that has the ability to be fertilized. That's a minimum requirement. But the line was drawn right short of that. So while we have the technology right now to generate these primordial germ cells from both XX and XY, the decision was not to allow fertilization to move forward, and therefore, for now, all these experiments are on hold until the discussion moves forward. I think that's a great example about how science has to move step by step with ethical and social consideration and not get ahead of it and not to try to impose the will of a scientist as the only truth that can guide the decisions we make about where do we come from, our reproductive rights or technologies. And we can discuss that more in a topic that's tangential to science. From the 3D side, we and others have the ability to generate blastoids. So this is before gastruloids. This is the ball of cell, the soccer ball and the tennis ball inside. And now you can generate these by the thousands, and you can convince yourself that they are the real thing. And now a whole new world of looking at symmetry breaking when just a ball of cell begins. And this is really, really recent. So 2 or 3 papers that's published. One of them from our side is in review. And something interesting happened. Again, remember T0 and time and day 14. These cells, again, have been encultured or embryonic stem cells or RUS2. You assemble them into 3-dimensional blastoids. And again, tens of thousands of them in any given wells. And we start looking at markers, and sure enough, I cannot tell you day 15 because I don't have T0, but day 5 is when these cells were isolated, and 10 days after differentiation, so it's 5 plus 10, would be around day 15, I guess, if the timing was in synchrony. And what we see is a symmetry breaking within this ball. One side is different than the other side. As you see these molecular markers. So we're generating symmetry breaking in these balls at day 15, kind of expected. But now we have the system to go back in time. So 15, 14, 13, 12, 11. And by doing so, something unexpected again happened. We start seeing symmetry breaking at day 5 plus 7, which is day 12 of human development, if that was, again, in synchrony with natural development. And you can see that the cells are breaking symmetry already. This is a marker of symmetry breaking, and one side, a few cells in one side become different than others. So that was weird. It gave us the impression that perhaps gastrulation does not start at day 14, but perhaps at day 12. So we went back to natural embryos from donations from the IVF clinic. And sure enough, Ricardo, the scientist in my lab, did the experiment using the same markers. And for the first time, we can conclude that human gastrulation starts at day 12, not 14. And that line was already too far when it was drawn in the 1970s. And we should have known. We should have known, because if you look at the frog embryo, before the sign of gastrulation and symmetry breaking happens, the molecular markers get segregated. The cells look identical, as I showed you, but one side is different than the other side, way before the morphological signs occur. And this is what you're looking at. These are the two cells here that just went symmetry breaking in a ball of cell. And this is the earliest time in our own gastrulation that I've ever seen. And if I take you into that drone again and put you inside of that structure, you would see these two daughters who suddenly are different than their neighbors in that tennis ball inside of the soccer ball. So this is how it begins. If the logic of the day 14 was based as we cannot cross human gastrulation, therefore it has to stop at day 14, that might be revised if you want to consider molecular markers. And these are the two cells for the first time. And so we will see what the reaction to that is. I think it's going to make us rethink about the origin of symmetry breaking, the formation of the anterior posterior axis, the separation of the head and the tail. Okay, so this does not come without a price. Here's the infernoid in 2 slides following this. There are punishments. Every time you do this, it takes a special kind of scientist not to follow the path of less resistance. And as I said, 25 years working on how you make a tadpole should have been sufficient to continue on doing that. I'm way past tenure by then. 5 years after Rockefeller, I was already a professor. So why go the extra mile if it's painful and full of punishment? I think there is something about science that drives us toward the truth, with the respect of all the rules. It goes back to Galileo, you know? It's not that the sun is not turning around Earth, it's the Earth turning around the sun. If you want to put me in jail and punish me for it, maybe, but there is a driving force behind the truth. And that's strongly influenced by political, not only by social, history, religion, or culture. It's strongly influenced by politics. I give you 2 examples in the United States. This is the State of the Union by George Bush in 2006. This is 2 weeks or a month after the human mouse camera was published as the first evidence that human embryonic stem cells have the ability to generate entire structures and perhaps entire embryos. And this is his declaration.
A hopeful society has institutions of science and medicine that do not cut ethical corners and that recognize the matchless value of every life. Tonight, I ask you to pass legislation to prohibit the most egregious abuses of medical research, human cloning in all its forms, creating or implanting embryos for experiments, creating human-animal hybrids, and buying, selling, or patenting human embryos. Human life is a gift from our Creator, and that gift should never be discarded, devalued, or put up for sale.
So this, as you can imagine, created a very strong reaction from everywhere, including Rockefeller University, which immediately took steps to detach from public funding to continue that research. My lab was literally separated in 2 parts. We could not take a pipette from one side to the other. We had to change our lab coats going from one side to the other. Every single piece of equipment had to be marked, identifying that the source of funding was not coming from the NIH or any kind of public source. There was reaction to that. California stood strong. CERN got up and said, you know, there is nothing wrong by using public funding to do research that would save human life. that it is important to balance moral and ethical values. By the way, people get confused when they talk about moral, they really mean ethics, and when they talk about ethics, they really mean morals. But one should balance this kind of consideration, which is all respected and respectable, with the benefits for the society. One should not take over the other, and there should be a debate as to which aspect and how can we balance these considerations without offending anyone. So the next round of authority, here is President Obama in January 2009, presidential executive order number 1.
We will bring the change that so many scientists and researchers, doctors and innovators, patients and loved ones have hoped for and fought for these past 8 years. We will lift the ban on federal funding for promising embryonic stem cell research.
So this kind of turbulence and wave will continue, and it does continue, as you know. Science has to steer its own way toward what it is acceptable in the conscience of a scientist and the society that the scientist is serving. Mass media is the other influence. Some people get really insulted by experiment. This is the Daily Express in London after the Nature paper with the human bird experiment was published. Abomination. So we used to use that word a lot in the lab for the weeks after this was saying, like, this omelet is an abomination. And then at the same time, Scientific Forum, this is Science in 2016, the breakthrough of the year, People's Choice. And the in vitro attached embryo is competing with some serious competitor. Ripples in space-time, experimental validation of Einstein's theory, comes second to the human embryo in a dish. And I think there is a reason for that. I don't think it's a coincidence. I think our thirst and our drive to understand where we come from is as big in the space in which we identify ourselves as an individual, in the infinitely small space of where we come from, compared to the infinitely large space that surrounds us. And I think there is a reason this attraction is difficult to quantify, but I think it does exist, and I think it will continue to exist, and we're very proud to be part of that. It's my last slide with my Andy Warhol to make you appreciate again how beautiful you are in so many different colors. We all start with the same shape and the same structure, and then we end up who we are sitting in this room. This is the team that contributed to my work and the source of funding, and I'm very grateful for your attention. And again, my apologies for passing that line on the sand. Thank you very much.
Thank you. So we have time for some questions. We have people that are ready to pass microphones for any of you that might have one. Does anybody want to lead us off?
When were you here last, Ali? When were you here last?
When I was here last?
About a year ago?
Yeah, about that.
This is a phenomenal talk, really fantastic. But can I ask you a non-science question?
Of course, if I can answer.
One of the things that is great about science is getting young people into the field. And we need those new young minds. You obviously came from a very wealthy background and had all the privileges and got to go to Berkeley and to Harvard and now at Rockefeller. What do you feel about young people who didn't have all the privileges that you did and think that maybe they can't enter this field?
That's a great question. I think the word privilege is a word that needs to be defined more than...
No, you know I'm joking about, I know your background.
Sometimes it's difficult to tell, but yeah. I've been bitten too much by too many different... When you get bitten by snakes, when you walk on branches, you get scared. But there is a very important point in what you're saying. I think privilege is driven not necessarily, but what you think that you want to do instinctively. To a large extent, it's a little bit like an embryonic cell developing within the context. It comes with the experience of the ecosystem. How far away are you from the edge? How do you recognize yourself? How do you measure that distance? It's all part of the journey. I come from a very unusual, I'm very lucky to be alive. I'm even luckier to be in your company and the education that I had. And maybe that's why I feel like life is a privilege that cannot be wasted by not doing something that really matters. And if you have the opportunity to do it in an academic environment, and if you have enough people who are willing to follow you, I think it's a mistake not to do it. So to me, it's a natural instinct.
Do you mind telling the young people, though, a little bit about your background and how you got into science, just so that they understand that someone from your type of background made it to the, very top?
Yeah, so I was born in Tehran, in Iran. I was lucky enough from a very, very young age for my father, who was a clinician, to take me infectious disease in his lab and showing me how to look down a microscope, at the time a single lens microscope, how to distinguish gram-positive from gram-negative bacteria. And somehow I got incubated into this environment. It's a little bit like my sons I was telling you about, how once you're into this, it's difficult to detach. But then a series of completely unexpected events, I guess symmetry-breaking events, occurred. The revolution started in Iran. We were relatively a comfortable family, as comfortable as a medical doctor can be at a very young age. Just started to buy an apartment, and I remember the first Volkswagen Bug our first car, and the first time we had a TV coming, that was great. It was like heaven. And then the whole roof collapsed. And everybody had to do their own things. The first thing my father did was to try to send us out, because the war was starting with Iraq and the southwestern border with Saddam Hussein attacking Iran. Kids were recruited at the age of 12 to run over the minefields, to clear it for the soldiers. And instinctively, you could just sit there and do it. So I was the first one out, followed by my sister. Then my younger brother came. He went through a lot of trouble. He spent a lot of time in jail because of that. But because he was a medical doctor, his services were needed at the time. And by miracle, I just came out of that situation. I lost most of my friends in high school. I lost my nearest member of my family, which was my cousin, same age as me. And that marks you. It marks you in ways that changes the way you look at life. And again, the privilege of being alive. And what are you going to do with it when you consider that you should have been dead long time ago? Maybe that's a driving force. Thing came back. I went to France. I went to school. I got my degree. I was in French school when I was a kid in Tehran, so that was my other language. By completely random coincidence, ended up in the US with my brother, who was 11 years old back then. Didn't speak a word of English. I still barely do. You notice all the mistakes I made. I came as an immigrant and a political refugee. It took me 3.5 years to get my paperwork together. 3.5 years. And again, by complete coincidence and luck, somehow I could get a temporary visa. I never got the political refugee status because there was still issues with Saddam Hussein in Iraq at the time. But that was good enough for me to apply to graduate school. I got to UC Berkeley. I got great mentors, great teachers. I learned from them. Harvard followed as a postdoc. I had 5 papers in graduate school. Still have the record, I think, there in about 4 years. Doug Melton immediately recruited me for a postdoc. I did only 2.5 years of postdoc. That was the neural induction pathways of how do you make a brain by default. And then Rockefeller recruited me under Thorstein Wiesel, the Nobel Prize for the president of Rockefeller. And since then, it has been like a dream, to be honest with you. I still look back at the time and where I come from, and I have to tell you, as to me, it's just completely, it's like magical almost. So doing this kind of thing kind of brings me back to where I come from. As I said, it's kind of like mirroring your past, remembering where you come from. And it manifests itself in this kind of work.
So a young kid who loved to play soccer
Yes.
managed to make it.
Yes,
Yeah,
absolutely.
I think that's a great story, inspiration.
Thank you. It's embryogenesis.
Well done, Peter, as usual, with his questions to draw out our speaker.
I thought you were going to go for the genetic signatures.
Do we have any others from the audience?
Yeah.
Can you tell us what is the mechanism by which the cells determine the distance? How do they know where they are located? And also, how do they know the shape, which is also you alluded to,
Yes. Yes.
without basically having the brains?
Yes. So this is, information processing is not only an attribute of neural networks in the nervous system, it can be reduced to the molecular levels. Self-assembly and self-organization is the essence of DNA replication, as you know, and transcription and translation is the essence of phage assembly or viral assembly. There is a mechanism that allows the cells to realize that they can be more than the sum of the parts, and that comes by communication. Up to these experiments, and again, I should have known, every embryo, a frog embryo, has a given shape. There is the outside world and what is contained inside. We had a discussion just before the talk. This is something that Claude Bernard defined as le milieu intérieur, the inside world versus the outside world. The distinction of where that edge is something that naturally should have been part of the equation for any embryologist. It's not only an issue of embryo. Every single tissue has delineation and boundaries, even liquid tissue like blood. So this idea that somehow this was neglected in terms of fate determination and patterning, with hindsight, is mind-blowing to me. Of course, there has to be a mechanism by which the cells recognize their edge. The fact that this comes from human embryo models rather than fish or frog or birds is also mind-blowing. It was right in front of me all the time, in front of everybody. So how many other physical, when I talk about light stimulation, the way biological systems such as the plants respond to cycles of light, these are all, should not be surprised anymore. The way they do it now is something amazing. In the case of edge sensing, there are two components. The first one is edge sensing is mediated by the localization of the receptors that get the signal. If you look at the colony from the side, so it's a circle, but if you look at it from the side and you get a cross-section, optical cross-section, and you ask, okay, so I'm adding a ligand, a single ligand, and suddenly everything self-organizes. You ask, well, where is the receptor for this ligand? It turns out that the receptor is localized within a ring right at the edge, at the apical side of the cell. As you move toward the center, the localization of the receptor becomes basolateral. So it no longer is responsive to what's going on from outside. You can actually prove that. You can grow those things in a transfilter system. You can present the ligand from the top or from the bottom. If you present from the top, only the ones from the edge will respond. If you present from the bottom, every single one of them will respond. So edge sensing comes from apical subcellular localization of the receptors. But this alone cannot account for self-organization because you can use trickeries like constitutively active receptors that will fire in the absence of ligands. You can use these trickeries and you still don't get the kind of refinement of self-organization you will see in size-dependent manner in a circle or a triangle. So there must have been something else. And when you ask, well, what is this something else? It's a reaction diffusion mechanism, the way it was theoretically defined by Turing in the 1940s, where by just very simple, he was interested in how a zebra makes its pattern. Is a zebra a white animal with black stripes or a black animal with white stripes? And so this is a very legitimate question. And in order to address that, and patterns like this exist everywhere, in zebrafish, in all animals, in us. If you look at your skin is not a homogeneous tissue. We're actually zebras. And so his theory was like, you only need two components for that. One is somehow figuring out how far away you are within a limit. And the second one is this concept of reaction diffusion, where from the limit comes something that inhibits a given signal. And so imagine if the source of a signal is here and is moving forward, that signal itself induces its own inhibitor, kind of self-censors itself. And its own inhibitor moves much faster than the ligand. So as this is progressing, the limit of progression is defined by the boundaries of the inhibitor. And you can tune this in a mathematical modeling. You can actually predict what would the size of the stripes be, depending on how fast the inhibitor would diffuse. And it turns out it's the same mechanism as in the zebra that makes the radially symmetrical pattern. He predicted it before any experimentation. That's the genius he was. You know, he is the first one who cracked the code on the Nazi communication for the Allies to be able to win the war, the father of the first computer, one of those geniuses that is not recognized enough, in my opinion. But he predicted it, and that's exactly what it is.
So my question had to do with the pattern that you imposed on the cells to get them to grow in certain structures on the plates. So how did you get them to grow, say, in a perfect circle or a circle with a hole in the middle or triangles, straight lines, stuff like that?
Very good. Okay, so cells can only attach to given substrates, and you can force them to attach to substrate, or you can force them to be repulsed by a basement to which they don't like to attach to. In this particular case of the microchip I showed you, is a UV filtering. So you generate a filter that you put on top of your microchip. You radiate, and then you lift the filter. And so only the parts that have received that signal that activates this thing called extracellular matrix is the base to which the cells can attach. What you do is actually extremely simple. You make the mask on the cover slip, you lift the mask, you put the cover slip in a small Petri dish, you just put the cells in it, and you gently mix, and then you let them sit. You pull the microchip out of the plate, and you put it into a new medium and a new plate, and everything is the pattern. The cells are attaching to the patterns that you impose on them. The size can be changed depending on the filter you make. You can actually generate microchips that have circles, squares, hexagons, triangle, all in the same. On the light induction mechanism, I showed you stripes of different widths and little stars you can make. You can write things. For Valentine's Day, I made one.
And so like, once you define that, it's set and stays for the rest of their differentiation?
Yes, and that's a very good point, because what happens if you change the shape dynamically? You know, we have been thinking about this a lot. In three-dimensional matrices like the 3D embryo I showed you, you can exercise stiffness and pressure. Like, how much do you allow this thing to expand? But we haven't done that in the microchip context. And that would be a great thing to do, because it would not only tell you how does a cell measure its distance vis-a-vis an edge in a static manner. We have to remember that edges in biological systems keep on changing, including, or especially, in the case of the embryo. So this dynamic readout of the edge, I think, is the next big question to address. I don't know the answer, but certainly we need to start thinking about it. That's a great question.
Thank you.
Peter has another question.
Regan, there's one more in the middle here. Sorry. Oh, go.
So you've commented on using light to establish patterns, but could you comment also on whether you can use it to entrain these embryos in vitro for circadian rhythms, for example, and how carefully you can recapitulate patterns of methylation and demethylation, imprinting epigenetic phenomenon occurring during this stage in normal embryos?
Very good. So we're now going from spatial and architectural imposition of patterns to molecular imposition of patterns. So one can interpret epigenetic marks, such as methylation, as establishment of patterns at the molecular level. But also related to the previous question, these are very dynamic. So doing it and undoing it, I think, is part of the way things work. If you had something that was very rigid, you would not allow changes in shape and in form. You want it to be rigid enough so self-organization happens, but you also want to maintain some plasticity. And again, we're talking before, during the hors d'oeuvres and the wine, the plasticity is extremely important. Consider yourself a frog in a pond, okay? So the female has laid 1,000 eggs, and the eggs are dividing very quickly. They're making a ball of silk, but you're still in the pond. So you want to keep the ability of when a fish comes and nibbles at you, and you lose a chunk of your sphere. If you're too rigid about, being dependent at the edge, you're done. You want to keep enough flexibility to say, this is my edge, I recognize my edge. But if something changes with that edge, I will have enough plasticity to recover. So I think things like epigenetic marks or cellular memory, I didn't have time to show you, but to a large extent, fate determination at the cellular level in the embryo, certainly in a human embryo, depends on the history of what that cell has seen. So you can give it a program, and that's translated by epigenetic marks and methylation. If I take a cell and I expose to, it's like the piano keyboard again, to a given rhythm, and then I wait X amount of time, and I give it another rhythm, compared to the same cell that has not seen the first rhythm, their reaction to it would be night and day. And the molecular basis of chronicle or maintenance of memory as you move forward, and again, we should have not been surprised maybe by this, is essential. You can only be what you know or what you have gone through. That defines you. It's relevant to how I that end up here. To a large extent is based on what you went through in life, most of it without necessarily choosing. The cell does the same and wants to make sure that while doing that, is plastic enough to overcome things that are perhaps unexpected. The translation of it is imprinting of the genome. Memory, cellular memory, is based on imprinting the genome. So I hope that kind of answers your question.
So I think we'll squeeze in one last.
So you've been talking about gene editing. You've shown layers and colors and symbols, and you coded them to each other. If you're gene editing, what exactly are you taking out? Are you eliminating a color, or?
Great question.
Is it that simple?
It's very simple, actually. Okay, well, you just tag something. It's like wearing uniform in a soccer game. So you can track it. You can see what the number is behind the shirt, and you can say, this is the one I'm going to track. In this kind of genome editing program, we put different fluorescent probes, and those shining in red or in green or in blue, depending on how you activate it, and we track the cells. These markers are supposed to be neutral. They're called lineage tracers. They go back to the early 1900s. The first experiments was done by the German School of Embryology by Otto Mangold, who was the husband of the person who got the Nobel Prize for Spearman. He wanted to know, where does the progeny of a given cell go? If you have two cells, is this one going to give rise to something else and this one to something else? And of course, we didn't have, nobody knew what DNA was at the time, in 1924. So the idea was great. The way he was doing it is to use a rusted nail, because he would just mark with rust. And so if you have a white frog embryo, all you have to do is to figure out where the color goes from what you just marked with. Later on, it became dyes, like the kind of dyes used in textile, indigo or Nile blue, where you would just touch a cell with a very small pipette or a glass or whatever you could get small enough, and those lipophilic dye will insert themselves in the membrane of a cell. So now this one will be blue and this one will not. So all you had to do, if you're a frog, if you're doing this experiment in the frog, is to see what are the blue cells in the tadpole. CRISPR-Cas9 genome editing in tagging is a very sophisticated way of things that were done hundreds of years ago. But it aims at the same. With much higher resolution, I want to track exactly where the daughters are going. That's what the purpose.
Oh, really? All right, one more. Just right here. Reagan.
How does embryo cell can treat type 1 diabetes?
Diabetes?
Type 1.
Yes. Okay, so the embryo makes decision as to what kind of grandmother to produce. You have these things called ectoderm, which are the grandmas of skin and brain and spinal cord and those derivatives. Then there is a group of cells, mesoderm, that give rise to blood, cartilage, bone, and descendants are going toward that organ. Pancreas comes from a group of cells called endoderm. So endoderm, if you consider a frog embryo, the north pole would be ectoderm, the south pole will be endoderm, and the ring in the equator will be mesoderm. So the cell sitting at the bottom of the sphere, in the case of an amphibian, decides how to give rise to all these endodermal derivatives. Endodermal derivatives are things like gut, pancreas, liver, all these internal organs that most people don't find as sexy as the brain. So, but that's where it comes from. And diabetes represent a functional abnormalities. Type 1 occurs very early during development or postnatal development, but it's downstream of the establishment of the fate by the grandmother. It happens downstream in the daughters of daughters of daughters. It is a serious problem, of course, but the origin of it can be traced very early, like most other diseases, by the way, I think, personally. Because we don't have enough genomes to accommodate for all the functions of 25,000 genes. It's like a piano with 25,000 keys, and we have infinite number of functions, biological functions. Look at the change of shape we go through. It's as if I was turning into an elephant in front of you. So we don't have enough information in my genetic code to accommodate all of this. So what I do is I recycle. If one of the keys goes off, then you generate a problem. In this case, diabetes in the endoderm. In other cases, maybe Huntington or autism, obviously. So these are the things that you have to be careful. There are some arguments about, is this part of plasticity or not? You might argue that, again, you don't want to keep things very rigid. We consider some diseases because they're diseases based on what we recognize as not being normal. But what if Huntington was actually an evolutionary advantage? If you look at the speed of development in the neurons in this system, a Huntington background differentiates in speeds that are much faster. Maybe that's one of those traits of random variation upon which Darwinian selection occurs. If you don't create variation enough, how do you know you're going to select for the best? So we consider short-term problems, obviously, that are sad, as aberrants, but they can also be considered as an attempt for evolutionary fitness. I'm not saying diabetes type 1 is one of these, but it doesn't escape my attention that there has to be some room for allowing things to be outside of the normal if you're going to generate things that are more adaptive.
So with that, if you'll join me in thanking Ali for his lecture tonight. And again, on behalf of UCI and the Stem Cell Research Center and myself as director, thank you for coming and joining us this evening. Have a wonderful 2023.